All right, welcome back everyone. It is the Water, Wetlands, and Watersheds Seminar for Fall 2020. It is November 18th, and today we're lucky enough to have Dr. Amanda Spivak with us. So before we get started, are there any announcements, any events coming up? Anyone got anything going on?
No, we'll probably have one more planting day out there at Airport Road, but it's not likely to be till early December based on the holiday next week and everything. But I will, that normal email will go out.
Okay. All right. Well, then, with no further ado, let me please introduce Dr. Amanda Spivak as an associate professor in the Department of Marine Sciences at the University of Georgia in Athens. Before coming to UGA in 2019, she was an assistant and associate scientist in marine chemistry and geochemistry at Woods Hole Oceanographic Institute in Massachusetts. She has her doctorate in marine science from the College of William and Mary in Williamsburg, Virginia, and her research focuses on coastal ecology. Her overall research goal is to understand ecological and geochemical processes in estuaries and coastal wetlands, and also how that's associated sort of with the global carbon cycle with global climate change from human disturbance. And her research uses innovative geochemical tracer approaches. So we hear about isotopes today and cool field scale mesocosms and lab scale work, as well as landscape experiments to quantify carbon pathways, transformations, and fate. So with that, Amanda, the floor is yours.
Great, thank you very much for having me today and for the invitation. Oops, I think I'm sharing the correct screen. There we go. So I'm really honored to be invited to participate in the seminar series, and I appreciate everyone spending a little bit of their day with me today to hear some more about some of our research on carbon dynamics in a New England salt marsh. I want to start off by acknowledging my Sharon Luck. Whoops, sorry, I was trying to get the pointer. There we go. Sharon Luck, who is a PhD student in my lab, and much of the data that you're going to see today she produced for her dissertation. And of course, science doesn't happen without collaborations, and I'm fortunate to work with some really great folks, including Kathy Todd Brown there at University of Florida. And so the overarching goal of this project is to characterize the effects of decomposition on soil organic matter composition and stability under both natural and disturbance settings. And we're keeping an eye here towards refining current assessments of carbon dynamics in marshes, as well as future predictions of marsh sustainability. And we're focusing on marshes because these are valuable but shrinking ecosystems. So salt marshes are a globally distributed habitat, but they account for a very small fraction of the world's land surface area. And the green shading on this map shows a global distribution of marshes. And you can see they're concentrated along coastlines. And this also happens to be where roughly 40% of the world's population lives. So this puts tremendous pressure on marshes. And it's estimated that 50% of marshes worldwide have either been degraded or lost. And in some areas, regional estimates are as high as 90%. Now, when we lose marshes, we're not just losing the habitat, but we're also losing the ecosystem services they provide. And that comes with a dollar amount of around $6 to $42 billion per year. Some of the most valuable ecosystem services that marshes provide include protection of coastal communities from rising sea levels and increasingly frequent and more intense storms. And we've certainly seen that this year. So marshes provide these services in part by gaining vertical elevation each year through the process of soil accretion. And they do this through largely two main mechanisms. So the first is that when the flooding tide comes in, it brings particles that are denoted here in these little stippled symbols. And these are generally mineral particles that are brought in by rivers. And as this material moves over the marsh flat, the marsh grasses themselves slow down the water velocity, and these particles fall out of suspension to the sediments and eventually become buried and contribute to accretion. The other main mechanism that contributes to vertical accretion is the annual growth of below ground roots. So these two mechanisms combined allow marshes to keep pace with sea level rise and provide these ecosystem services. Now, a few years ago, Nat Weston really nicely demonstrated that riverine delivery of suspended sediments is declining to marshes, and he focused on marshes along the Gulf Coast and East Coast of the US here. And the declines in suspended sediment delivery are denoted by these different red circles. So at the same time we're having a reduction in suspended sediment delivery, we're having an increase and relative sea level rise rates, denoted here by these blue circles. And so what this means is that marsh survival is increasingly depending on organic matter burial and preservation. And marshes are really good at producing and burying organic matter. Peat deposits can be hundreds to thousands of years old. And the sufficient preservation of soil organic carbon is really the outcome of two processes: the really high rates of production by marsh grasses and slow rates of decomposition by soil microbes. And we generally conceptualize decomposition in marshes by proceeding through one of two pathways, either this fast cycling pathway in red or this blue slow cycling pathway. This fast cycling pathway, organic matter decomposes in the matter of years to decades, and so this material doesn't contribute very much to long-term elevation maintenance and gain. The slow cycling pool, however, turns over on timescales of centuries to millennia. And this is really the component of soil organic carbon that's critical for supporting carbon storage and elevation maintenance. Often the material in the slow cycling pool is characterized as lignin, and it's thought that it's sort of recalcitrant or resistant to degradation. But we know from recent evidence in the terrestrial literature that decomposition is limited more by environmental constraints on the microbial community rather than an intrinsic property of molecular recalcitrance of specific compounds. The implication for marshes then is that they're likely preserving a broader mixture of organic matter than just lignin, and that these compounds are associated with different reactivities. So disturbances that change environmental conditions can have the potential to accelerate decomposition and the loss of long buried carbon. And this is just a really dramatic example off the coast of Massachusetts of waves breaking on a relic marsh. And you can see the exposed peat faces here, and even some that are being washed out to sea. So accelerated decomposition can lead to not just loss of carbon from these systems, but eventually ecosystem subsidence and ecosystem loss. So the questions that we wanted to approach in this project, well, we wanted to understand the sources, reactivity, and turnover times of this slow cycling carbon, how decadal scale disturbances affect soil carbon reactivity and composition, and then how do disturbances that accelerate decomposition affect the ecosystem carbon budget. And we address these questions in the salt marshes of the Plum Island Ecosystems Long-Term Ecological Research Site, which is on the North Shore of Massachusetts in Rowley. This is just an overview of the LTER domain. And this is the slice of marsh that we're working in. And more specifically, we're focusing at these 3 starred areas. And we picked these areas because they represent both a marsh platform and a pond. And in this design, the marsh platform represents an undisturbed natural condition, and the pond represents a decades-long disturbance where we have accelerated decomposition. We also picked these 3 sites because they're all located in the high marsh, so they have very similar elevations and they have very similar tidal flooding patterns. The ponds also differ in size, so we have a small, medium, and large pond, and they host different abundances of macroalgae and submerged aquatic vegetation, mainly rubia. And so we have nice ecological gradients there. And we focused on ponds as a disturbance because in many systems, their spatial extent is rapidly increasing. And I just have a few examples here. So this is from Rhode Island, from a paper by Watson and others in 2016, where they compared aerial extent of ponds between 1939 and 2011. Here's another example from the Blackwater marshes in Maryland, where you see the arrows are denoting places where we have increased hydrologic connectivity and then changes in ponding extent. And then from Cape Cod, this is the Great Barnstable Marsh. And between 1973, I've highlighted 2 different pond complexes that have expanded. Now, ponds are natural features of marshes, and they can exist for decades before draining and recovering. But with rapid rates of sea level rise and certain hydrological management practices, we're seeing expansion of shallow ponds in many salt marshes. Ponds typically form following disturbances, and this can be something like increased waterlogging or rack deposition that causes the marsh grass to die back. And then the ponds progressively deepen over time through decomposition of the underlying peat. So we can use ponds as a natural decadal scale experiment for evaluating disturbance impacts on soil organic carbon. And in our system, ponds can be really prominent features of salt marshes. And so I've put up, to illustrate that, I've put up an aerial image of our study site and mirrored it here with the distribution of different habitat types in our study area, denoted by LIDAR. And what I want to draw your attention to are all these bright blue dots. What these represent are individual isolated ponds. And in this slice of the marsh, they represent 8% of the land cover. But in Pi itself, we have over 12,000 individual ponds, and the ponded area has been increasing in recent decades, mainly due to a decrease in drainage density. So the ponds that we're focusing on this study began to form between 1965 and 1978. And at the time of sampling, this represented between 36 and 49 years of disturbance. And we're able to determine that the ponds formed in this time frame based on historic aerial images. So we addressed our 3 research questions by looking at carbon dynamics in marsh soils and in ponds. So we collected meter-long cores from the marsh and analyze them for organic matter thermal properties, composition, and carbon isotopes. And we constructed lead-210 geochronologies to understand changes over time. We also collected 50-centimeter cores from the marsh, and we conducted the same analyses. The rationale for this design is that we know from our historic aerial images that these ponds used to be vegetative marsh. So in theory, soil organic matter at this depth, at this elevation, should be quite comparable to organic matter composition in the nearby marsh site. But at the surface of the pond, these soils have been exposed to changing environmental conditions, so change in submergence, a change in oxygen due to the populations of benthic microalgae. And this should represent our accelerated decomposition environment. And so we would expect to see changes in soil organic carbon reactivity and composition that are coincident or that reflect these changes in disturbance environment. And like I said, this work that I'm presenting is actually that of Sharon, who's a PhD student in my lab. And so I'm very shamelessly plugging her data and results. We addressed our 3rd question about to look at how ponding affects the marsh carbon budget by looking at carbon dynamics in ponds themselves. So by characterizing the metabolism of the whole pond and then of the sediments. So we'll start off by looking at our long core data by comparing properties and processes in paired soil cores between the marsh and the ponds. And we'll start with our soil bulk properties. And so just to orient you to my graphs, we have this elevation axis. So this is going down core in our sediments. So this would represent the surface of the marsh, and this would represent the bottom of that marsh core. And then in blue here, we have our paired pond cores. So with bulk density in our rooting zone, we have relatively constant bulk density and relatively constant total organic carbon in this rooting zone. But below the rooting zone, we see a decrease in carbon content and an increase in bulk density. And what this represents is a loss of organic matter due to microbial decomposition, followed by compaction and dewatering of the soil column. In our pond cores, we have a lower bulk density And what this is reflecting, we think, is the collapse of the marsh grass roots and then water infilling the existing pore spaces. And so that would be contributing to a higher water content and a lower bulk density. Total organic carbon content, as in percent, is quite similar throughout the profile between the marsh and the pond. And then when we multiply carbon percent by bulk density, we see very similar profiles below the surface of the pond, between the pond and the marsh. We see lower carbon densities at the surface of the pond compared to the comparable elevation in the marsh core. And so the alignment of these trends going down core, of the marsh and the pond going down core, provides some support for our comparative approach and the slight difference in the surface horizon. But these are just bulk properties. They don't tell us a lot about composition. And so we used a couple different ways to get at what's there and how reactive it is. And one of the first things we did was use a variety of lipid biomarkers. And here I'm just showing some of our hydrocarbon data. And what we've done here is taken this ratio of odd chain to even chain hydrocarbons. And this roughly represents a ratio of vascular plant to algal organic matter. And in our marsh core, we have a relatively straight profile. But in our pond core, at least at depth, we have overlap between our marsh and our pond. But at the surface, we have a relatively higher ratio. And so this suggests we have relative accumulation of vascular plant material relative to algal material. And so this provides a little bit more support for the idea that the pond sediments are decomposition is proceeding in a way that it's decomposing the labile algal material first, and this residual, more refractory vascular plant material is getting left behind. The next thing we did is we characterized soil organic carbon thermal reactivity and carbon sources using this technique called ramp pyrolysis oxidation. And essentially what we do in this is we take a soil sample and we put it in a reactor, and then we heat that to 1,000 degrees in the presence of oxygen. And then that gas then goes through a catalyst. And so we have conversion of these reduced compounds to CO2, and we measure the CO2 on an infrared gas analyzer. And so we get thermograms or profiles that look like this. So as we increase temperature, we have organic matter that's burning off and being converted into CO2. And what we can take away from this is that organic matter that burns off at lower temperatures, we classify this as more thermally reactive, and organic matter that's burning off at higher temperatures, we classify this as being more thermally stable. The next thing we can do is we can collect the CO2 as it's burning off and analyze it for its 13C and 14C composition. And so we can do this across the thermogram, and we can overlay metrics of thermal reactivity with carbon source and carbon age. And we did all this up at the NOSAMS facility in Woods Hole, and this inset is just a picture of the instrument itself. So we'll walk through some of these profiles. So what we're looking at here is we're looking at profiles, thermograms, from 4 different horizons in the marsh core, and we're going down core and elevation, so just a little core graphic to guide that. And what we're seeing here in this dark line is the average CO2 evolution over temperature for our 3 marsh sites, and the gray area represents the variance. And what we find is that we have 2 very distinct peaks. We have a low temperature peak and a high temperature peak separated by this trough in the middle, and that these peaks persist as we go down in the core, even though the shape and the overall height of the peaks may change. Looking at the ponds, our pond cores are at a lower elevation, so this is the surface of the pond, and this corresponds in elevation to this horizon in the marsh core. But we see a very similar pattern, right? We have 2 very distinct peaks, one at a lower temperature, one at a higher temperature, and they persist going down core. And so we see we have different organic matter representing different thermal reactivities in these soils, and they're persisting. But looking at peak wiggles is a little bit qualitative, and so the next thing we want to do is convert these data into indices of reactivity. And we did this in two different ways. The first thing we did is we took this ratio of the areas under the low and high temperature peak, and then we calculated the thermal activation energy required to combust the organic matter in these samples. And so again, we're going from the top of our cores down into the sediments. And what we see in our marsh core is that we have a higher peak ratio at the surface, and then we have the loss of that low temperature peak, and so a decrease in our ratio. And coincident with that, we see the opposite pattern in our thermal activation energy. So thermal activation energy is lower at the surface, and then it increases with depth. And so we see this progressive shift towards increased stability, thermal stability with depth in the marsh sediments. Looking at our ponds, we have a different pattern. So at the surface of our ponds, we have a lower peak ratio. This indicates smaller contribution of that low temperature peak relative to the high temperature peak. And we have a higher activation energy. But as we go down core, we have overlap both in terms of activation energy and this peak ratio between our marsh and pond cores. So this provides, again, a little bit more evidence that our design here is addressing what we think it is, that back in time, our marsh and pond cores were had quite similar organic matter, but this long-term disturbance of ponding is causing a shift in organic matter reactivity to be relatively more thermally stable. So the next thing we wanted to do was to look at the specific sources of carbon and the ages of carbon that are burning off in each of these temperature fractions or associated with different temperature fractions. And because doing isotope analyses on gases is quite time-consuming and expensive, we only did this at one of our representative sites. And this site, we have the marsh next to our largest pond, which is also our oldest pond. And so we're just going to walk through, we're going to overlay the isotope signatures on the backgrounds of our thermograms. And so our 13C signatures are overlaid in these red dots. And essentially, these dots represent the midpoints of the temperature fraction that we collected over, or that Sharon collected over. And we have three potential sources of carbon that are being buried in the sediments. So one, we have a autochthonous marsh grass carbon, and that would range in del13C values between -13 to -15. We have potential inputs from the marine side, which would be more depleted, so around -18 to -24. And then we have potential for terrestrial inputs, so from the riverine side, which would be even more depleted, so -26 to -30. And what you can see here is that all of our del13C values are consistent with marsh-derived organic matter. So that means that this marsh is primarily burying organic matter that's produced within the system. The other thing that you'll see here is that we have consistent variation in our del13C signatures across our thermogram. So in our lower temperature peaks, we generally have more depleted values than in our higher temperature peaks, and this becomes more clear as we move down core. And so there are a few potential things that could be driving this. So one is that we could have an artifact in the method, but because the shift in del13C values with temperature is not monotonic, we can rule that out. We don't think this is a kinetic isotope effect because the differences that we're measuring are larger than what would be predicted based on kinetic isotope effects. And so we think these are real differences in the del13C value of our CO2 fractions across these thermograms. And we think that these represent variability in the biochemical composition of the grasses. So Spartina alterniflora is around 70% cellulose and hemicellulose, and that has a del13C value of around -11.5. Lignin makes up a smaller fraction of Spartina biomass, and it has a more depleted value. And so looking back at our del13C values here, we generally have more depleted values in our lower temperature fractions and more enriched values in our higher temperature fractions. So what this is suggesting is that we might have relatively greater inputs of cellulose and hemicellulose represented in these higher temperature fractions, and relatively higher or greater representation of lignin in these lower temperature fractions. Now, this was a bit surprising, particularly since there's plenty of experimental evidence out there that's demonstrating that cellulose and hemicellulose degrade more rapidly than lignin. So this brings up an important caveat about this method, that thermal reactivity doesn't necessarily reflect bioavailability. And so we would need to think more about what this means in terms of what's actually available to the microbes and what they're using. But the other thing that we note as we go down core is that we have a general trend towards more enriched values and more similar values between our lower and higher temperature peaks. And what we think this is reflecting is the long-term effects of microbial reworking. And so, as you'll notice here, our deepest horizon is 75 to 77 centimeters. And so these sediments have been there for a very long time. And so what these data then are telling us are that even though these sediments have been there buried for a very long time, microbial decomposition has slowed, but it hasn't stopped. And so it's likely that we're still having decomposition at these very, very deep layers. So we also looked at the del13C values of our pond cores. And what you'll see here is that they overlap quite well with what we saw in the marsh cores. So one, this provides additional support for our comparative approach between the marshes and the pond. And two, it means that the ponds are primarily reflecting marsh-derived organic matter. Now, this was particularly surprising in this surface horizon. So like I said, the ponds are somewhere between 36 and 49 years old. And over that time, they've developed macroalgal, microalgal, and submerged aquatic grass communities. But we're not seeing those contributions reflected in these del13C values. And what that suggests is that that material is not contributing to deposition in the sediment, so it's likely being decomposed quite rapidly. So the next thing we wanted to do was to overlay age information, and we did this by looking at 14C. And so what you'll see now is these rectangles, and these represent the thermal fractions, the temperature fractions that Sharon collected the CO2 gas over. And on the right-hand axis here, this is the F14C value. So adding in 14C, we can now parse sources out a little bit more specifically. We can look at new marsh material, which would have a modern F14C value, and we can look at, distinguish that from old marsh, which would have a value less than 1. We can further parse out marine and terrestrial organic matter because we would expect them to have different radiocarbon signatures. And so this allows us to tease apart sources a bit better. So starting again with our top fraction here, what you'll see is our radiocarbon values are around 1 or a little bit greater than 1 in our lower temperature fractions. And then as we move to our more thermally stable organic matter, we're winding up with much older organic matter. And we see this in these top two horizons. And then the distribution of 14C values becomes more homogeneous with depth. So in our surface horizons here, our lower temperature peak represents this new inputs of marsh material. So this would be autochthonous recent production. And these older ages suggest that we're having erosion of creek bank material and redistribution and redeposition of that material throughout the marsh. We can calculate out how much carbon is associated with each fraction. And when we do that, what that suggests is that erosion and redeposition of old creek bank carbon is actually contributing 9 to 17% of the total carbon in these surface horizons. And so this is potentially important for a few different reasons. So one, when we think about quantifying soil carbon stocks in marshes, we generally think that we're only quantifying new production that represents, the past couple of decades. But what this is saying is that old carbon is actually can be a pretty significant part of that. The other thing is that as sea levels continue to rise and we have more erosion, happening, or it's likely that we're going to have that this is going to be more important become a more important mechanism in terms of marshes maintaining elevation and building vertical accretion each year. The other thing that we are able to do with this technique is by overlaying our thermal information, our 13C and 14C information, we can identify at least 5 geochemically distinct carbon pools. Now, this is a pretty big departure from how we generally think about carbon being buried in marshes. We generally think it's just the lignin fraction, but our data here is showing that it's actually much more complex than we've been historically thinking about it. We also looked at our 14C data in our pond cores. And again, we see really similar patterns between our pond cores and our marsh cores, in that we have generally values that are closer to 1 or slightly greater than 1 in our lower temperature horizons, and values that are much less than 1 in our more thermally stable fractions. And this would reflect inputs of older organic matter. Now, again, this was kind of surprising in our pond surfaces because these systems have developed their own plant communities. We would expect higher inputs of recent organic matter production in our pond surfaces. And this is suggesting that most of the organic matter is primarily driving from the marsh, and new production is not really contributing to the soils. The other thing that this indicates is that ponds aren't acting as a trap for organic matter that's being washed off, off the marsh platform. If that were the case, we would expect to see radiocarbon values that are around 1 or even greater than 1. So the next thing we wanted to do was to use these down core patterns in organic matter reactivity and concentration to calculate turnover times. And to do this, we used a depth for time substitution design in which we modeled our slow cycling organic matter as either a single pool, so this would be bulk soil organic carbon, and this would be sort of a traditional way of approaching it, as a 2-pool model. Then this would be based on our low temperature and our high temperature peaks that persisted throughout our soil cores, or a 5-pool model that reflects the combination of our thermal and isotopic data representing greater diversity of organic matter and reactivities in the soils. So we modeled these 3 different kinds of pools against natural geochronometers in the soils. And so for soils that have been deposited since 1900, we built age models using lead-210. And then for soils that were deposited before 1900, we extended our lead-210 constant initial concentration model and combined that with late Holocene rates of sea level rise and glacial isostatic adjustment. We only did this for the marsh cores as due to violation of steady state assumptions for the lead-210 models with the pond formation. And we had differences in our lead-210 inventories, which suggested some erosion. These models were based on equations described in Manzoni, by Manzoni and others in 2009. And really Sharon and Kathy really led this work. And you can find out more about their equations on Sharon's GitHub repository. But a few things about the models that this, the turnover of the bulk soil organic carbon in the first most thermally reactive pool are described by a first order kinetics. And then the subsequent pools are modeled as a linear series where a fraction of the decomposed material from the previous pool is inputted to the next pool as an additional input. And so we're starting off with our 1-pool model. So again, this is bulk soil organic carbon concentration. And so we have total organic carbon concentration on our y-axis, and then time in years from our natural geochronometers on our x-axis. The individual dots are our individual data points, and the line is the best fit model. And what Sharon's put up here is the 750 years represents the best fit model, and then she's put the range in parentheses. And so the best fit of 750 years is actually in agreement with some marsh evolution models. It's a little bit on the higher end, but it's consistent. Then we did our two-pool model, and this again is based on this lower temperature fraction. This represents the first pool in black, and then our second temperature peak, our higher temperature peak, representing the second pool, which is in gray. And again, we have a relatively faster turnover of this first pool compared to our second pool. And the second pool is turning over at a rate that's 49% slower than the first pool. And it's also 40% slower than this one pool model. So by adding in this complexity, we're showing that these different pools are turning over at different rates. The next thing that Sharon and Kathy did was that they created this 5-pool model that's based on our geochemically distinct fractions by overlaying that thermal and isotopic information. And so our first pool here in red decays the fastest, followed by the second and the third pool. But what I really want to draw your attention to are these fourth and fifth pools. And so you'll see that over time, these are actually increasing. And this is an important mechanism in terms of marshes maintaining elevation and building elevation and keeping up with sea level rise. And the accumulation of these stable pools is mostly due to slower turnover rates, as well as increased transfer over time from the preceding pools. Now, there's certainly a lot of variability there, and we certainly see really wide ranges in our ages here, or our turnover times here. And so what we think moving forward is that this two-pool model is a nice compromise, as it captures the different turnover rates of slow cycling pool. And it's a nice compromise between this more simplistic model and this more complex model. So through these approaches, we've developed some of the first geochemically based decay rates in marsh soils. But what about ponds? We know we can't apply the same approaches here because we violate the assumptions of LET-210 modeling. And so we took a different tack by looking at pond metabolism. And we did this first by looking at metabolism in the of the whole pond and the surface waters. And then we looked at metabolism rates in the sediments, and then looked at carbon sources used by sediment bacteria. And so because ponds are semi-isolated systems, we're able to use a free water approach to measuring metabolism. And this is based on diel swings in dissolved oxygen. And all three of our ponds had really dynamic swings in DO. They were supersaturated during the day, and they were hypoxic to anoxic at night. So we calculated metabolism for the 3 ponds that I showed you earlier, and we did this using this free water diel method. We constrained gas piston velocity based on coal and Caraco. We corrected our Schmidt numbers for salinity, and we calculated metabolism using an ordinary least squared method. So what you're seeing here is the outcome of those calculations. So we calculated rates of gross production, net production, and respiration for our 3 ponds during the summer and the fall, and during isolated and flushing tide stages. So for gross production, we had the highest rates in pond 1, where we had the most abundant plant communities, and our lowest rates in pond 3, where plant communities were more depauperate. We found higher rates of gross production in the summer compared to the fall, so reflecting senescence. When we look at net production, we found, so here a positive number means the system is net autotrophic, and a negative number means that the system is net heterotrophic. And so here for pond 1, which had our most abundant plant communities, it was net autotrophic in the summer and in the fall. Pond 3, where plant communities were more depauperate, we had, it was net heterotrophic in both the summer and the fall. And pond 2, which had intermediate plant communities, was autotrophic in the summer and heterotrophic in the fall. But what we're really interested in here are respiration rates, and we see a very different pattern. So here, respiration rates were actually quite similar across all 3 ponds, and they were a little bit higher in the summer compared to the fall. And so what this suggests, these similar rates across the pond suggest that respiration is largely decoupled from the plant community. And this is mainly being driven by sediment heterotrophs. So the next thing we want to do is to better constrain sediment respiration rates. And we did this by collecting sediment cores from the ponds and collecting the pore water at different depths and constructing pore water profiles to calculate diffusive flux rates. So we did this for a couple of different analytes, but I'm just going to show dissolved inorganic carbon on the top here and sulfate on the bottom here. So for DIC, a positive number here means an efflux from the sediment into the water column. And we had our highest rates in pond 1, intermediate in pond 2, and lowest in pond 3. And again, this is consistent with where we have differences in plant abundances, but we don't see any difference between seasons. So for sulfate, our diffusive flux here, a negative number means that we had consumption of sulfate. So this would represent sulfate reduction, and this was the main non-aerobic metabolism in these systems. And again, our highest rates were in pond 1, and our lowest rates are in pond 3. One of the other things you'll notice about pond 3 is that our error bars, or even the entire bar, are above our 0 line here. And so this represents either no sulfate reduction is happening, or that we have sulfide oxidation. And so an important point about these data is that they represent the net effect of both sulfate reduction and sulfide oxidation. So these are a really conservative estimate of sediment metabolism rates. The other thing that we found was that there is no difference, again, in sulfate reduction rates between the summer and the fall. And that was really surprising because we did see seasonal senescence of our plant communities. And we were certainly out there late enough in the season to collect a seasonal difference. Our ponds froze over, and we literally had to break the ice to get our cores. Made for a really long day. So our flux rates here in the sediments were really similar to shallow estuaries, but much slower than vegetated marshes. And these similar seasonal rates suggest that bacteria were not tightly coupled to the local plant communities. So this brought up the next question is, what are the bacteria actually using? And so to look at that, we compared the isotopic composition of bacteria to their 2 main carbon sources. And so in these systems, the main carbon sources would be either benthic microalgae, which live on the surface of pond sediments, or the underlying peat. And so in this top graph here, I've looked at the del13C value for polyunsaturated fatty acids, and these would represent benthic microalgae in these systems. And while we have variability across our 3 ponds and with the different tide stages, which is a really interesting story that's for another time, but most of these values are around -9 to -15. So if bacteria were tightly coupled to our sediment microalgal community, we'd expect to find the same pattern in our bacterial lipids, and we would expect to see overlapping del13C values, but we really don't. So here in this bottom figure are the del13C values for our branched fatty acids, and these would be indicative of heterotrophic bacteria. And what we find is that there are similar values across all 3 ponds that don't match our benthic microalgal lipid signatures, and that they're more depleted. So they're all around -14 to say -17 compared to the benthic microalgae. But when we compare those values to the Marsh SOC, so the bulk carbon that we looked at earlier, that was around -15. And then when we look at our specific carbon fractions, they range from -13 to -17. So what these data are suggesting is that the pond bacteria aren't tightly coupled to benthic microalgae, but they're likely decomposing a mix of organic matter. And so some of that probably is coming from benthic microalgae, but these data provide a mechanism for bacteria decomposing the underlying peat, contributing to carbon loss. So we next wanted to do sort of a thought exercise about the role of decomposition in pond expansion. So at a big scale, we have basically 2 main processes contributing to pond depth. One is accretion of the surrounding marsh, and the second is decomposition of the underlying peat. We know from our soil cores that the mean marsh accretion rates is around 2.5 millimeters per year, We know from aerial photos that our ponds are between 36 and 49 years old. So that means over the lifetime of the pond, the marshes gain an elevation of 9 to 12 centimeters, and this represents around 30 to 42% of the pond depth. In order to decompose the remaining volume of the pond, we would need a respiration rate of around 23 to 50 millimoles of carbon per meter squared per day. And this actually agrees quite well with our whole pond oxygen metabolism rates and our dissolved inorganic carbon efflux rates from our soil cores. So with our isotope data and with our rate data, we can come up with a very plausible mechanism for decomposition contributing to pond expansion and carbon loss in the system. So the next thing we wanted to do is to try to scale up and think about the role of ponds in marsh ecosystems. And I put this picture up here because you can see that ponds vary in sizes and shapes. And so it begs the question of how scalable are our rates? So in a paper that just came out a month or so ago, we published the results from trying to understand scaling effects of pond metabolism rates. And what you'll see here is that we calculated gross ecosystem production, net ecosystem production, and respiration in 6 different ponds represented by these 6 different dots. And they're color-coded according to different regions in the marsh. So dots with the same color represent areas of the marsh that were similar hydrologically, so they were supplied by the same tidal creek. And so we looked at 3 different regions of the marsh to try to get an idea of how well these processes scale. And so we compared our production, gross and net production and respiration rates, our ponds across area, volume, and surface area to volume ratios, and we see no relationships, which was a bit disappointing, but it means that it's going to be complicated to integrate ponds in the ecosystem carbon budgets. But one comforting thing here is that the range of rates is actually not all that broad. And granted, this is only 6 ponds, but we can at least use this as a starting point for estimating pond effects on marsh metabolism. So we took that step by combining our estimates of pond metabolism with published rates of production and respiration for the emergent grasses and soils in the system from Forbridge and Giblin, based on the land cover classifications from our LIDAR data. And when we do this, we find that accounting for ponds results in lower rates of net ecosystem production and respiration than if we didn't account for ponds. And so accounting for ponds actually lowers what our current estimates are for our ecosystem rates of production and respiration. So that was sort of a whirlwind tour of some of the different things we've been thinking about in terms of carbon cycling in marshes. And our main conclusions here are that the slow cycling marsh carbon is much more complex than we generally conceptualize it and is represented in ecosystem evolution models. That centuries of microbial reworking leads to an accumulation of old and thermally stable carbon. That erosion and redeposition is currently playing an important role in marsh carbon budgets in this system, and is likely to become increasingly important with sea level rise in the future. This decadal scale disturbance of ponding accelerates the loss of thermally reactive organic matter and results in accumulation of thermally stable organic matter. And we find evidence that bacteria can readily use this old soil organic carbon given favorable environmental conditions. And thinking about the system more integratedly, we find that pond expansion is reducing soil carbon burial and altering ecosystem carbon cycling. So with that, I'd really like to thank many of the people who are either involved directly in the research or provide a lot of guidance to my thinking, as well as our funding from NSF and NOAA. We had lots of support from NOSAMS, the USGS, and the Plum Island Ecosystems LTER. And I'd be happy to take questions.
Awesome. Thank you so much, Amanda. As always, we appreciate someone taking us from the scale of the soil core to the whole pond to the entire LTER. It seems like we've got a lot of speakers doing it this semester, which is amazing. And it's always great also to see the diel oxygen signatures used for metabolism as the Odom Center for Wetlands. We always appreciate that, aquatic respiration method. All right, so I'll open it up. Any questions from the audience that are here on Zoom? And I'll check the YouTube as well. Andrew, I see you're here.
Yep. Hey, hi, Amanda. This is Andrew LTER. I'm on the faculty here in Environmental Engineering Sciences, and enjoyed your talk. I've done a bit of marsh work in New England as a postdoc when I was working in Burtness Lab. And I'm not working in marshes so much now, but I've poked around enough to know in the Southeast that the marshes are a bit different, or in many cases at least, they're more like these sediment-based rather than peat-based marshes. And I'm just curious, because I imagine you're starting to look around in this region a bit more, basically how pond, dynamics, if they exist in the same way here, might differ based on that kind of difference in just kind of the physical structure and the sediment structure in those marshes. What your thoughts are on that?
Yeah, so the marshes down here are very, very different from what I'm accustomed to in New England. So at least in the Georgia coast, they're definitely more mineralogenic. And because of that, we don't really see much ponding. And so that's sort of interesting to think about, why we see ponding in some settings and why we don't see ponding in other settings. Certainly there's definitely ponding along the Louisiana coast. And they're seeing it, I think in some areas of the Gulf, but we don't get much of it here. And so, yeah, I mean, that's something that we're starting to explore, is to better understand carbon cycling and what are the processes that are maintaining elevation in the marshes down here.
Yeah, that's interesting. I mean, because I guess one thought I had when I was, as you're going through your talk, I mean, the ponds are these really nice, like lens and understanding marsh kind of dynamics. I think you do a really nice job showcasing that. But of course, the creeks themselves are obviously very dynamic, and they're interacting with the sediment and changing biogeochemical processes. And I'm just wondering, like, what your thoughts are on that. I mean, that all these marshes down here, even if they don't have ponds, have creeks, and it seems like you might be able to ask some of the same kinds of questions working in those creeks, particularly the ones that seem to be rapidly expanding. And I've collaborated a bit on some of those dynamics.
Yeah, so we actually am quite lucky. I have a new project that's getting underway with Giulio Mariotti at LSU, and we're looking at tidal creek migration. So he's looking at tidal creek migration from a much larger scale, in the modeling scale, and quantifying net changes in marsh extent over centuries. And then my role in that project is sort of understanding how marsh pergradation and retreat affects carbon cycling and the kind of organic matter that survives being resuspended and then redeposited elsewhere. Is it primarily terrestrial material that's surviving? Is it primarily marsh material? So what are the sources and the reactivity of that material? But yes, that's you see the pathway we're going on.
Yeah, stay tuned. Cool. Well, thank you. It was a really nice talk. Appreciate it.
Thank you.
Thanks, Amanda. We have a question from YouTube from Alexandra Bijak. She asked, do you think bioturbation plays a role in SOC in your marshes and ponds? I was intrigued by the same idea. So faunal interactions. Faunal
interactions, they're very important. In many systems, and our ponds, because they go anoxic at night and they're going anoxic for like anywhere from 2 to 8 hours a night, they're really stressful places to live. And so we do get some bioturbators. We actually get mummy chogs in the sediment, if you'll believe that. And we see shrimp and things like that. We don't have many fiddler crabs in the marshes up there, at least when we sampled. That could be changing. And we don't think there's a ton of bioturbation effects on the soil, because when we looked at the beryllium-7, which is a short-lived radioisotope, we don't see it below 1 centimeter. And so if there's a lot of mixing that's occurring due to bioturbation, it's really constrained to that surface horizon.
Yeah, interesting. So thinking just about, climate change and sea level rise as these major bottom-up sort of biophysical or physical drivers that are also leading to tropicalization. I know Andrew and Christine on the call both, looking at increased ranges for various organisms and how that might affect things. But I'll open it up if Mark or anyone else in class has a question. Oh, there's Christine on her walk.
Yes, I'm out getting exercise. I have one question, although and I'll do it quickly. Amanda, that was really nice. It was really cool to see all the different tools you have in your toolbox. So thank you for that. I think I'm even more excited to find ways of working with you now.
Great.
So this is very productive for that reason. I was intrigued by some of your results suggesting that like the lateral erosion of these marshes is really contributing to like the superficial deposition of what's there. And I'm wondering if in the, you know, Pi LTER, you're actually moving forward to couple those rates of lateral loss of the marshes with vertical accretion rates to see if there's actual correspondence between like the spatial extent of the marshes and how they're kind of peeling back from the edges and what you're actually seeing in terms of sediment deposition on the surface, and whether ultimately that deposition rate is sufficient to keep these marshes above, you know, above sea level.
Yeah, so that's a great question. So fortunately, Chuck Hopkinson has been thinking a lot about that, and he and some others had a paper that came out in 2018 where they calculated the contribution of old organic matter to the marsh surface, and they came up with really similar estimates. I feel like theirs was like 12% or something like that. It was in the same ballpark as ours. And so there are a couple different lines of evidence suggesting that, you know, the tidal creeks are eroding and widening, and that material, instead of being exported from the system, is redistributed across the marsh. And that is one mechanism that's supporting the marsh in keeping up with sea level. Thanks for tuning in on your walk.
This is the best way to listen to seminars.
We really appreciate it. Thank you so much for coming to give our talk virtually, and we'll hope to see the next set of results, maybe next semester or next fall. We'd love to have you back.
Great. Well, thank you so much for the invitation and everyone for taking the time.
Yeah, pleasure. Thanks, everyone. We do have seminar next week. Is that right, Sherilyn? We do have?
Yes.
Yes. Okay, so